Welcome to part 6. In this part, we'll discuss validation of these genetic tools. And a question that commonly arises, do estimated breeding values really work? We receive many questions related to the efficacy of estimated breeding values. People question their accuracy and whether the statistical methods used to calculate these breeding values really represent what's going on as far as the animal's genetics. Over the last several years, 2 studies have been done that focus on the validation of these breeding values, and I hope to provide some data here to support the fact that these breeding values do work, they are accurate, and they can be used with confidence to select sheep and improve the performance of those economically relevant traits. The first study I'll discuss relates to the validation of the fecal egg count estimated breeding value. The primary question being, does the fecal egg count estimated breeding value work as a selection tool for parasite resistance? In addition to that, some work was done working to better understand the mechanisms underpinning resistance or susceptibility to parasitism. This work was done at West Virginia University in collaboration with USDA and Virginia Tech. The project was done using Katahdin sheep at the Southwest Virginia Agricultural Research and Extension Center. The work shown here is part of a study led by Dr. Scott Bowdridge. So as I mentioned, part of this work aimed at understanding the mechanisms that control that parasite resistance and whether an individual is parasite resistant or more parasite susceptible. We know those genotypes based on the fecal egg count estimated breeding value. So we know what the genotype is based on those breeding values, and we know what the phenotype is. We know if that individual doesn't show resistance to parasitism or is indeed susceptible to parasitism. But what is the difference? What's the link between that genotype and that phenotype? And for parasitism, that is that immune response. There's a cellular and antibody immune response associated with parasite resistance. And this work aimed at better understanding what this immune response is and how that immune response targets that parasite. More information related to this will be discussed as part of the parasite management section of this course. For this study, 5 rams that were extremely low for their fecal egg count estimated breeding value were selected. In addition, 5 rams that were extremely high for their fecal egg count estimated breeding value were also selected. So in total, 10 rams were utilized for the project. These rams were randomly mated to use at the Southwest Virginia Agricultural Research and Extension Center in Glade Spring, Virginia. So the rams were randomly assigned to those ewes, half of the rams being very parasite resistant based on that fecal egg count breeding value, and half the rams being very parasite susceptible based on that estimated breeding value. Those post-weaning fecal egg count estimated breeding values along with their accuracies are provided here. So you see for our low sires, the average was negative 85% for the fecal egg count EBV. For the high sires, the average was 288% for the fecal egg count EBV. As far as the project timeline, lambs were born in mid-March. They were moved to pasture shortly after lambing and managed on grass with their dams until weaning. Starting in mid-May, fecal egg count collections began, and fecal egg counts were collected every 2 weeks for the duration of the project. Lambs were weaned in the first week of June and then managed on grass post-weaning for roughly a month. In mid-July, lambs were moved to West Virginia University, where they underwent artificial infection. So these lambs were housed in a feedlot environment and were artificially given the parasite infections so that we could better understand that parasite infection in a more controlled environment. That fall, lambs were then harvested and adult worm counts were determined at slaughter. Long and short, when we evaluated the fecal egg count of these individuals over the grazing period, the fecal egg count on lambs sired by low fecal egg count rams was lower than the fecal egg count on lambs sired by high fecal egg count rams. So selection works. The fecal egg count EBV does work. By utilizing low fecal egg count sires, we were able to decrease the fecal egg count on the resulting progeny. In addition, there was a lower deworming requirement for those low fecal egg count sired lambs. In addition to validating the breeding value, We also better understood how that fecal egg count fluctuates in the time around weaning. The timing of that fecal egg count collection is critical to the accuracy of the data collected. I have labeled here when weaning occurred the first week of June. You see, leading up to weaning, the fecal egg counts were relatively low, but the high fecal egg count sired lambs did have a higher fecal egg count than the low fecal egg count sired lambs before weaning. After weaning, for about a month, that fecal egg count showed quite a bit of variation and fluctuated. And actually, for one time point, the low fecal egg count sired lambs had a higher fecal egg count than the high fecal egg count sired lambs. So we see a lot of variation. However, what's important to note is by the time we reach about 3 to 4 weeks post-weaning, those fecal egg counts segregate and diverge as we would expect, where the high fecal egg count sired lambs have higher fecal egg counts than the low fecal egg count sired lambs. So again, the fecal egg count EBV works. We do get that divergence that we would expect based on that breeding value. But we need to remember that in that period around weaning and the stress associated with weaning, we may see some variation in that fecal egg count. Those lambs are under stress and their immune response may be compromised during this time, resulting in some fluctuations in fecal egg count and maybe some greater parasite susceptibility in some individuals. And so that's important to keep in mind. If you're interested in collecting fecal egg counts and utilizing these genetic tools, we need to make sure that we collect that fecal egg count at or before weaning, or we need to wait at least 3 to 4 weeks after weaning so that we can ensure we have an accurate fecal egg count number that's representative of that individual's true genetic merit. So that's the period that we're trying to avoid, that period starting at weaning or shortly after weaning for about 3 to 4 weeks after weaning is when we want to avoid collecting those fecal egg counts. Second project I'll discuss was called the Leading Edge Project. The goal was to validate the growth and muscling EBVs in our Suffolk rams. In addition, this project looked at the economic impacts of selection based on growth or muscle in Western range production systems. This project was also a collaboration between the American Sheep Industry Association Let's Grow program, the University of Nebraska-Lincoln and Dr. Ron Lewis's lab, the National Sheep Improvement Program, as well as Superior Farms, Mountain States Rosen, and Allflex. More information about this project is provided at the link below. This project utilized 42 Suffolk rams in a Western range production system. 15 of these rams were non-NSIP rams. These were labeled or will be labeled as industry rams. These rams were not a part of the National Sheep Improvement Program and did not have estimated breeding values. As far as the NSIP rams go, 13 rams that were selected were a part of NSIP and had extreme or higher estimated breeding values for post-weaning weight. So these rams were selected for post-weaning weight EBVs. These are labeled as the weight rams. And then 14 of the NSIP rams were selected for eye muscle depth EBV, or these rams had an extremely high EBV for eye muscle depth. This is labeled as the muscle group. So there was 3 treatment groups. We had our industry group, our weight group, and our muscle group. As far as the project timeline goes here, in the fall, rams were joined with approximately 1,100 ewes in multi-sire mating groups at Mickle Brothers Sheep Company in Spring City, Utah. In April, lambing began and 1,491 lambs were tagged and DNA samples were collected to validate parentage. In early summers, those pairs were moved to mountain grazing for the duration of the summer. And in September, approximately 1,100, just over 1,100 lambs were weaned and shipped to a feedlot in Idaho. These lambs were then fed at that feedlot. And over the course of that winter, as lambs reached a similar weight, they were harvested by Superior Packing in Dixon, California. Results of the DNA parentage showed that 12 rams sired 10 or fewer progeny. So there was 10 rams that hardly sired any progeny. There was 2 rams, though, that sired over 100 progeny each. And so with this DNA parentage, we're able to evaluate or better understand how some rams have a greater breeding capacity and may be able to breed quite a few more ewes and have quite a few more progeny than other rams. And more work is being done to understand why that might be. the majority of rams sired between 10 and 40 progeny. So the majority of rams sired between 10 and 40, but we did have a few rams down on the extremes that may have sired close to 100 or maybe sired 10 or less. So these lambs were evaluated for growth and muscling traits. That level of performance was compared back to the ram's EBVs. That's summarized here in this table. So we have our EBV category in the first column. We either have our weight group, those Suffolk rams selected based on post-weaning weight, EBV, and then we have our muscle group, those rams selected for that muscling, eye muscle depth, EBV, and then the difference between the two. Here we're primarily focused on weight at weaning. So then we have in the next two columns their weight EPD. So these is simply divided by 2. So we can have the level of performance we would expect to see in the progeny, the relative difference in performance between these two groups. So are their weaning weight EPD is 4.6 for the weight group, 1.9 for the muscle group. The difference here is 2.6 pounds. For post-weaning, their post-weaning EPD for the weight group is 9.2, for the muscle group 4.2, the difference here being 5 pounds. So based on their breeding values, we would expect lambs sired by the weight rams to be heavier than lambs sired by the muscle rams. When the progeny weight was actually taken, so at weaning, all of the lambs were weighed. The average weaning age, remember this is a Western range production system, so these lambs are weaned at an older age. The average weaning age was 161 days. Lambs were all weighed at that point. Lambs sired by the weight rams weighed on average 108.6 pounds. The lambs sired by the muscle rams weighed on average 104.1 pounds, the difference being 4.5 pounds. Because lambs are weaned at an older age, this is more representative of the NSIP EBV for post-weaning weights. Remember, the post-weaning weight EBV is taken between 90 and 150 days of age. These lambs, because they were weaned at an older age, we need to look at and utilize the post-weaning weight breeding value. In this case, the difference in post-weaning weight breeding value between the weight and muscle groups is expected to be about 5 pounds. In reality, when we weighed these lambs, the difference in their weight at 161 days of age was 4.5 pounds. So here in this case, that breeding value allowed us to predict the difference in lamb weight within a ½ pound. So by utilizing those weight rams versus the muscle rams, we increased our lamb weight by 4.5 pounds. You multiply that across the number of lambs that were produced through this project and through all of those matings and the market value of those lambs, and there's certainly some economic benefit there. So these breeding values are accurate. They do support what we say they support. The level of performance that we would expect to see based on the breeding values is accurate and is representative in lamb crops when they're evaluated. More validation work will continue to be done, but these two projects I think were fundamental in setting the groundwork for explaining and validating not only the fecal egg count breeding value, but also the weight and muscling breeding values as well.